Okay, welcome everyone. We're going to talk about open science for a while. As you can see, the title of the talk is "Do you speak open science?" And if not, here's why you should, because open science has become a very important thing in science, and especially in the life sciences, where we are kind of at the forefront of this. And we'll talk about the aspects of open science in all its glory in the next few minutes. So in a way, when I started doing open science, because I was kind of active in this field, I sometimes felt like the knight on his horse there, fighting off a dragon. And the whole idea was that in order to make the science open, we had to kind of fight for that, right? It was a big thing because a lot of people didn't want science to be open. A lot of people had interests in not making it open. And in order to liberate the data in distress, we needed knights in shining armor to take care of business. However, as time progressed, I realized that this was more like fighting windmills, because especially now, the battle for open science has been fought. It's not like as if we still need to fight for open science. It's rather that open science is now more or less the standard modus operandi, and it will continue to grow as a standard modus operandi. And as a result, the battle has kind of been fought. So rather than tell you about why we need open science and why you should have open science, we'll spend more time on how you can do open science and what you can potentially do with open science. Because I think that's the bottom line, and the take-home message is that a lot of people have fought for open science, and essentially they've won. So while that fight may have been glorious, in all honesty, the fight is kind of over, and the real challenges now are to make it all work in practice, which is the responsibility of all of us to do some things, and we'll talk about what these things are. And then secondly, to also make this work for people. And to make things work in open science, we have to then use all of this data that has become available to do interesting things with, because that's how you extract the value from open science. So for those of you who want to read some more about this, together with Dr. Paola Masuzo, who was a very talented postdoc in my lab, we've written a manuscript that is called "Do You Speak Open Science" as well. And as you can see, that's a preprint. It's indicated quite clearly at the top, and it's not peer-reviewed. So we didn't submit this to a journal and have it undergo peer review. We just wrote kind of a white paper, if you like, on how to implement open science. And we just made that available to the community. This manuscript has seen a lot of interest. I mean, the screenshot is very old, so the views are limited, but right now it would be a whole lot more. You can see that this was actually a screenshot from 2017, I think. And it's actually influenced policy at the level of the European Commission as well, which I'm really happy about. And it crops up here and there. So occasionally people send me photos of meetings at, for instance, the European Commission in Brussels, where suddenly this manuscript shows up on the projection. So apparently it's not a bad place to start reading about open science. And of course, it's open, so it's really available, so you can go and read it for free. Okay, so I'll first talk a little bit about the many reasons that exist for open science, and which are the good reasons and which are rather the bad reasons, because not all of the reasons are equal, I think. Then we'll talk about open data, which is kind of the closest thing to what you guys are doing right now, because you're doing experiments, you're creating information, you're acquiring data, and so that data at some point needs to be open. Second thing you will do with that data is write a manuscript about it, and then you probably want to think about open access of those manuscripts, of those papers that come out. Then we'll talk about open code, which is also important for all of you who write code. And then finally, I'll talk a little bit about open peer review, not too much, because that's probably the least developed aspect of open science. It's also a very detailed aspect of open science. It's something that mostly impacts scientists, and very specifically scientists, but nevertheless, it's worth talking a little bit about that. And then I'll end with a final call to action, essentially trying to get you guys to realize that all this open science business is creating an enormous amount of opportunities for you guys as well. So let's start with the many reasons for open science. Now, for many people, open science is something they have to do. Yeah, so you will remember that I mentioned that we kind of won the battle on open science a while back. And as a result, we now see, say, the European Commission's Horizon 2020 framework, and undoubtedly also their next framework, Horizon Europe, that essentially has very strict guidelines on data management, on open access of the publications, and so forth. So the whole open science idea has been built in to the main European Commission funding scheme for science. It's not just the European Commission. You can see a lot of other funders at the bottom. So ERC in the middle there obviously is also part of the European Framework for Science funding, so they have the same policy. But you see big institutes like NIH, the National Science Foundation in the US, EMBL, and then two private funders as well. And they have actually been pioneers in this. The Wellcome Trust is highlighted, and the Bill and Melinda Gates Foundation. And these are the two biggest private funders of science in the world. And both of them have taken very, very severe stances towards open science. So they really want open science to succeed. Maybe most notably, Wellcome Trust and Bill and Melinda Gates Foundation nowadays prohibit you from publishing work in journals that are not completely open science. We'll come back to that, but that fundamentally means that if you get money from the Bill and Melinda Gates Foundation, that work can no longer be published in Nature, it cannot be published in Science, it cannot be published in Cell. You have to go elsewhere with your work. They prohibit you from publishing in these high impact factor journals because these are not fully open access. So they take a very, very strong position here, which is kind of leading the way for where other funders will likely be going in the future. So the other people who carry a stick are precisely these journals. So this is an editorial from Nature Biotechnology, obviously a big journal in the life sciences. And this is already from quite a few years back, and they essentially made an editorial stating, if you now want to publish in our journal, you have to somehow make your data available. So your data has to be open. This, of course, is a very interesting way of forcing people to make their data open, because if you want to have your manuscript accepted in Nature Biotech, before they will finally accept your manuscript, you will need to make your data available. And so, of course, everybody scrambles to make their data available. There are also some issues with this last minute scrambling to make data available. We'll come back to that in the open data section. But it did help because it changes everybody's mindset. It suddenly tells everyone that sharing data is not just a good idea, it's mandatory now. And so from these two angles, you can see that open science is becoming far more important and essentially has been very well established. However, this is still that people make you do this, right? So is this, it's effective, but is it nice? Probably not. If you're forced to do something and you don't want to do this, then that's for you a bad thing. So this is coercion, if you like. Then the other reason that is very often quoted for open science, and the one that I dislike the most, is that open science is meant to catch people perpetrating scientific fraud. So the idea being, if your data is open, we will catch you if you do nasty stuff that is not okay in proper science. Now, I give you one example that was happened in 2016. There was a paper published in Science, and it concerned a study of microplastic beads and how they affected fish larvae. So microplastics, as you probably all know, are being found everywhere. So it's a major pollutant, and people care about whether or not this is harmful for life and therefore humans. And so this study focused on fish larvae. And the conclusion was that, yes, these microplastic beads are really bad for fish larvae. And as a result, this was picked up by all the newspapers all over the world. This was a big item. It says microplastics are going to be very bad for you and they're everywhere. So this is an environmental hazard we should really care about. So far, so good. Now, the problem was that these scientists actually did their research in a marine research station somewhere on the coast of Sweden. And there were other researchers present at that marine research station who, when they read the paper, thought, we've never seen them do any experiments like that at the marine station. So where does their data come from? We have no idea whether or not this is real. So they actually asked for the data. And then you can see on the right-hand side here in blue, they were asked to produce the data that they built their whole case on, and they told science, oh, but they stole our laptop. And this laptop is the only place in the universe where our data was kept. So we can't give you the data because we lost it. So that's a big red flag. Yeah, so if you don't have any data, and there are people out there who claim you don't have any data because you didn't do any of the work, it's going to be very difficult to prove that you in fact did the work and had the data at one point or other. So then there's a very long story behind this. This is worth reading the whole story. With these pointers, you can find it online. But fundamentally, in a second level of investigation, not the first level, but the second level of investigation by a review board, they found some very serious problems. The first one was that there was no ethical approval for the experiments. Now, mind you, if the experiments actually never took place, I don't think that's too big a problem, because you don't need ethical approval for experiments you haven't done. However, since they can't even produce the original data that they should have acquired, they're essentially using this against the credibility of the work. And then finally, it was also completely unclear how these experiments were carried out, because there's no lab notebooks detailing the experiments. Nobody witnessed the experiments apart from the two authors. And so fundamentally, they said, this is not okay. The paper should be retracted, and the paper was retracted in full in 2017. And of course, 2 careers were ruined at the same time. So it's a typical example of potential scientific fraud, most likely scientific fraud. And people say, if only we had open science, we would have caught this, this case earlier. Well, to that, I tend to say, it's not necessary to have open science to catch this kind of thing. First of all, this case was cracked not because of open science, but because people in the marine station felt it was important to point out that no, these people probably didn't do those experiments, therefore this is probably bogus. So people got caught not because they were open, but because other people took their responsibility. And this is very, very often the case. So people who cheat in scientific papers tend to be caught out, not because their data is open, but because other people either whistleblow on them, or because people find weird things that shouldn't be there in the papers. And the fraud detection, therefore, does not necessarily rely on open data. In fact, if you are very good at fabricating data, you could have open data and people still wouldn't notice that you are cheating. It depends on how sophisticated a cheat you are. So as a result, I'm not entirely happy with making this link between open science and fraud detection. Yes, it can help a little bit, but fundamentally, open science is not there to enable fraud detection. It may be one of the tools you can use in a process of fraud detection, but really open science is a whole lot more than just trying to find out nasty scientists. So I think that's really a very negative way of positioning open science, and it's definitely not the core business of open science. So what should we focus on then? Why should we be doing open science? These are the positive reasons, the ones that I think will motivate people rather than scare them off. First and foremost, as a scientist, I think that making your work accessible to all the other scientists is one of the coolest things you can do. Why do I think this is so important? Well, for one thing, I like to get many references to my work, because that means my work is relevant. And if I make my work accessible, well, there is a better chance that people will pick it up. Also, if other people reuse my work, I get really happy, because I've made somebody else's life better. I've allowed somebody else to jump a little further in their research because of what I have done. So I think that's a really important reason. We're not in science because we're selfish people. We're in science because we want to help humanity. And this is a very nice reason to make your work accessible. Second is that, and I kind of covered that already, that if your work is accessible, other people can progress faster. So imagine now we are, of course, in the middle of a coronavirus outbreak. If people would sequence the virus and then sit on the sequence and say, now I'm the only person in the world who knows the sequence of this virus, so I have this huge competitive advantage, and I'm going to score a few more high impact papers than my colleagues. I mean, this is not how we make progress. We make progress by throwing everything out there and having as many minds as possible look at the problem in order to crack it as quickly as possible. And if we do this iteratively and everybody contributes everything that they have found in addition to what has already been found in the public domain, then we can jump so much faster and so much further in our work. The next one is that, of course, if you do your work in a little island and never talk to anybody about this, the usefulness of your work is very, very limited. It may give you a good feeling. It's not going to be able to help anyone. So this is essentially an economic argument. It means that society invests a considerable amount of money in scientific research. And so it is our responsibility to try and make sure that that research gives maximum benefit to society. And the only way to maximize the benefit to society is by making everything available to society. So society pays us, and the product of our work paid for by society is given back to society. That's kind of a simple economic argument that I think makes a lot of sense. Now, this penultimate one is a tricky one. It's tricky because a lot of people in the experimental side of the life sciences have some issues with this statement, but nevertheless, fundamentally, it is very true. These days, the data that you acquire from your experiments tends to have a potentially much longer shelf life than whatever you wrote in your manuscripts. That's because your manuscripts are essentially interpretations of those data. And while they could be perfectly correct and perfectly usable, very often the data contains a lot more information than what you extracted to write that manuscript. So almost anything in the life sciences today, and in fact over the past 20 years or so, has become high throughput, so you get lots of data, and high content, which means that whatever you record much more data than you're ever going to use. Say you sequence a whole genome, and you're going to annotate some open reading frames, and the rest of the DNA that you don't know what it's for, you're not going to look at. So now you've sequenced a genome and you've annotated open reading frames and you've written a paper about that. But anybody who studies, say, enhancers, or something completely different like a molecular clock could be super interested in the non-coding parts of your genome. But maybe that's something that you've never thought about, that you would never do, but that data is there. It's part of what you have acquired. It's just a part that you didn't work with. So being able to make that data available and have other people do other things with your data that you would have never thought of, that you would not have been capable of, or that you wouldn't even have known was possible, makes it that all data in the life sciences nowadays tends to have a much longer shelf life, a much longer useful lifespan than the manuscript you wrote about it. It's kind of a sobering idea because we tend to like to think about our papers as the ultimate expression of us as scientists, which is kind of true. But ultimately, unfortunately, it's fun, but it's not the most important thing. The most important thing is the data that you acquired from those experiments and making that available. And then finally, and this is, I think, the most exciting bit about everything with open science, is as a guy who runs a group of researchers who work only with public data, I can tell you that open science allows such groups, allows everybody who cares to become very, very creative with all this data that is available. And it stimulates what I call revolutionary research, the kind of stuff you would never be able to do on a single data set. Because there are certain things which will only be possible if you have, say, 10,000 data sets rather than 1 or 2 or 5. And this particular thing, this revolutionary research that can encompass the big picture of all data, that's the kind of stuff I think that for young scientists should be the most enticing. Because anything you do in that space, you will likely currently be the first one to ever do something like this. That makes it scary because you're the first one and you don't really know how this should be done. So everything is kind of, you have to play it by ear. You have to be very careful. But at the same time, everything you do will be a breakthrough of some sort. And right now we're in a place where this open science thing is sufficiently mature to allow this kind of research to happen before it became mainstream. To put that in another way, a slightly more negative way, if you like, is if you don't jump on this bandwagon now, you're going to lack a very important skill going forward, because 10 years from now, it's not going to be revolutionary anymore. This kind of stuff will now be standard. And if you're not capable of keeping up with that standard way of working, then you're going to be out of date. So it's changing the way that we do science, that we can do science. It's not taking away and moving somewhere else. It's essentially creating new opportunities that didn't exist before, but that will become very important going forward. But taking together, I think all of these reasons give you a very positive view on open science, a way in which this is sharing, this is creating opportunities, this is extracting maximal value, this is essentially allowing us as scientists to go forward as best we can. Right, so hopefully I've convinced you that open science, there are many reasons, some are better than others, but fundamentally there are very many good reasons. Now let's talk about the 4 distinct aspects of open science, and we'll start with open data. That's as close to the experiment as you can get. So there is a very nice set of principles outlined already for many years, and they're called the FAIR principles. They stand for, as you can see, findable, accessible, interoperable, and reusable. So let's have a quick look at what these things mean. So findable, as you can see by the magnifying glass, means that these data can easily be located by anyone. If you make a beautiful data set and you chuck it in a zip file on your private server at the university, the probability of anybody ever finding out about that data set is practically 0. Especially if you don't index it in Google, so not even the file name can be found. On the other hand, if you take say a sequence of the coronavirus strain that is now ravaging the world, and you put that in the NCBI database, everybody's going to find that because that's where people go. They go to GenBank to find new sequences from, say, a virus. So they can just type the name and poof, they will find it. So that's a very important aspect. Make sure that when you contribute data, other people can actually pick it up. However, that's not enough. So if you make your data findable, you also must make it accessible. So what does that mean? So when I started out in my field to try and create an open science system, the problem that I encountered was that people actually made their results available, but they did so in PDF tables. Very long PDF tables, some of which were Excel sheets that were converted to PDF. So you could try to recreate an Excel sheet from that, but that would be extremely difficult. Very often the markup would go completely bananas and you would get a really bad Excel sheet back. And some of these, and I kid you not, were actually scanned versions of printed out Excel sheets. So they were essentially images. And these images, you could try to OCR them, but it was very, very complicated. These data sets were actually easily findable in the sense that they came along with the manuscript. So if you found a paper that you thought was interesting, and you said, oh, I can look at the supplementary information, it was a link away. So you could find it then, but the problem was it wasn't accessible. Because we're talking here about, say, 7,000 identifications of peptides. And if I have to manually type that over, it's going to take forever. So I'm not going to do that, especially not for 20 or 30 or 40 papers. So accessible means getting that data out there in a way that people can immediately use. So for instance, a tab delimited file would not be a bad idea. Then everybody can read that. That's super easy to read. And in fact, you can probably be sure that they can read this, 300 years from now still, because it's a text-based format. It's easy. Which actually seamlessly brings me to interoperable. Interoperable means that you choose a findable and accessible set of data that is in a format that allows other people to reuse it with little hassle. So imagine that you make your own little zip file available somewhere on your server. And then in that little zip file, you have 7,000 tab delimited files, each of which contains some aspect of your data. And you might even have written a little document that says, oh, but file number 1 contains X. File number 2 contains X linked through Y. File number 3 contains Z. File number 4 contains Y linked to Z, and so forth and so forth. So you can make this very complicated relationship, and then people should figure it out. That's not actually that's accessible, but it's not very interoperable. This means I cannot plug this into, say, R, or into Excel, or into some other tool without actually having to go through a lot of effort. So instead, it makes sense to make something available as one giant, say, TSV file or some standard relational database format that I can then very easily import in my own tool, be that a Tableau data analysis tool or R, or maybe SPSS, or I could load your relational database schema in my Postgres or in my MariaDB or in my Oracle, right? So this whole idea of interoperability means give it a format that other people will be very happy and will be very convenient for people to reuse. And then ultimately, of course, your data, why do we do all of this? So that your data is reusable. And then the question is, so what else do we need except for findable, accessible, and interoperable to ensure reusable? And that essentially boils down to metadata. So I'll talk about metadata in some more in detail later, but fundamentally, if you make an Excel sheet or a tab delimited file available that has a bunch of numbers in it, and each in columns, and each column is named column 1, column 2, column 3, column 4, and column 5. I have no idea what these numbers mean. I don't have units. I don't have a description of where the number comes from, of any process that might be applied to them. It could even be that all these columns are related, but I would have a very hard time to find out that, say, column 2 is the square root of column 1, which is then to the 3rd power in column 3, and so forth, right? So the point is that data becomes reusable when you can understand the actual individual data points in the data set. And again, that is very tricky. It's more tricky than the previous 3. And I'll show you later on from experience why I can say that. Now, of course, a lot of systems exist, and this is just a first sampling. There are very many more of these that implement these FAIR principles already. Now, the nice thing is, if you make your data available through one of these resources, they will actually guide you to make sure that your data is fair, because they have these operating procedures in place for how to deposit data, how it should minimally be annotated, what it should look like, which formats it should have, so that by the end of the submission process, you likely will have findable, accessible, interoperable, and reusable data. And this is a very important aspect of open science, is that it should be low threshold. It should be as easy as possible for you to submit your data to such a resource. Very importantly, if you're at the start of collecting your data, it makes sense to spend 1 hour, 2 hours to go and look at the repository or resource where you might want to submit that data and just check the kind of stuff they make you do. Because that means that you will be aware of what you will need at the end before you start. And then you can make a good track record of all the things that you will need later on, which usually is not a lot, but it's super annoying if there's this one little setting that they really would like you to report, but you forgot to write it down. Because then what do you do? So it makes sense to have a good idea of, okay, I want to submit a structure to PDB at one point or the other. Know what PDB will be asking of you at the end of the trip, so that you know at the beginning what your checklist should be, and then keep track of everything. And then the submission process at the end is going to be peanuts. It's going to be very easy, okay? Right. So fundamentally, the sharing of data requires 3 building blocks, regardless of how you look at it. And these 3 building blocks are minimal reporting requirements, which is essentially provide all the information that anybody else would ever need to correctly interpret the data. So this really is what kind of stuff should you be talking about to correctly describe your experiment? Yeah. So for instance, where does the sample come from? What did you do to the sample? If you ran it through some machine or other, what were the settings on that machine that makes sense for other people to know? If you did anything to your data after it came off the machine, like normalize it or fix something or remove outliers, document that because people need to know about that, etc. Okay, so this is really a description. These minimal reporting requirements are very often compared to a methods section in a paper, because the methods section in a paper is kind of a minimal reporting requirement set. It tells you what happened, how you got at stuff, and how things became to be the way they are. A second thing here is controlled vocabularies. Controlled vocabularies are very important because we want to make sure that everybody understands everybody else. You guys are a very diverse group of people. I have a very diverse lab. So all of us understand that we need to have a common language in science. So we use English. We speak English with each other because now we can understand each other. If everybody would use their own language, it would be extremely difficult to communicate with each other, or we would have to know a lot of languages. The same thing goes for data. If everybody just chucks down whatever they think is a good description for a given data point or for a given procedure, then we won't know what we're talking about. And a very nasty example in the life sciences is yeast to hybrid, because there was this time when yeast to hybrid was written in 700 different forms, not counting all the spelling mistakes you would encounter. And so for a human, you kind of could figure out that they're talking about yeast to hybrid. But if you were a computer plowing through data, you would not really understand that these were the same things. And everybody would use their own abbreviations and would use their own way of reporting the simple term yeast to hybrid. Now, what people have then done is they said, you know what, we'll make a controlled vocabulary. We'll make one standard term for this, yeast to hybrid. And then we'll annotate it with as many synonyms as possible so that even through text you could find the synonyms, but we'll also abstract the concept with a number and we'll remember the number. And then we can make links between numbers, so we can say that yeast to hybrid, that number, is a kind of protein-protein interaction technology. And you can see that in the tree structure that is indicated here. And that tree structure actually allows you to find ever more fine-grained subtopics of a given topic. So not only do you get unambiguous annotation, you also get this annotation in a wider context. Okay, so if you make your own yeast to hybrid variant, it is a type of yeast to hybrid, which is a protein-protein interaction technology. This is really cool for the whole findable thing, because now I can say, look, I want to find all protein-protein interaction methods that have been employed on this type of sample. And whether that's yeast to hybrid, or it's tandem affinity purification, or it's something completely different, I will find all of them because all of these have registered themselves as being one of these. And I'm asking for this, which automatically allows me to find these guys. So that's a really nice benefit of a controlled vocabulary. Not only do we understand each other, but as soon as there are links that show hierarchies behind the terms, we can actually do very powerful queries on the data as well. And then finally, we need standard formats. So standard formats, we talked about that. You want to make sure that everybody can read your data. So if you come along with a proprietary format, that is going to be very difficult for other people to read. So at some point or other, in many of the omics fields, it was problematic because vendors would store the data coming from their instruments in their own proprietary format, and you would not have access to that format unless you bought the software. And the problem was they wouldn't sell you the software. They sold you the instrument and the software came on the instrument. So if you're a bioinformatician who wants to help out a colleague, you had one option, and that was you could buy the instrument, which you would never use, but then at least you could read the data format. Obviously, people were super unhappy about that, and this got fixed. So nowadays, it's easier to get at data, but nevertheless, standard formats that are defined by the community, not by a single vendor, and that tend to be very open, not very complex. So they tend to be text-based because text can be read for a very long time. are very important in making sure that everybody can read stuff. So this is interoperability, but at the same time also accessibility. And so you don't need anything special to read it, and you've made it such that it's easy to read this whole thing. Oh, by the way, occasionally you will see P. Matsutso show up here in the bottom right. That's when I stole a slide from Paola Matsutso, who thankfully also allows her slides to be shared, but we'll come back to that later. Right, so I promised to talk about metadata. I'll tell you here about some metadata from the PRIDE repository. PRIDE stands for Proteomics Identifications Database, and this is the open science system that I built from my field, which is mass spectrometry-based proteomics. So I wanted to make a database where people would share their mass spectrometry data, and so that we could reanalyze it. Because again, it's high content, high throughput data, and there's a ton of stuff that can be found there that would not be accessible from individual data sets, but it is there if you have many data sets. And in fact, it's been a really big success. It's now mandatory to deposit your data there. So that's why the growth is so strong. And the other thing is that a lot of people are reusing that data now. So it really became an ecosystem of data exchange, which is fantastic. However, this side here on the left has all of the instrument annotations in the database. And as you can see, the most popular instrument is unknown. Now, if you think about this, this is mass spectrometry data, which means that, you know, the key thing here is the mass spectrometer. All of these mass specs are different, or most of these are quite different. And so you want to know which instrument the data came from in order to interpret it correctly. But the vast majority of people really could not be bothered to annotate even that. And because even though it was the key technology that they employed, it's one thing that you have to specify, too much hassle. It gets worse because here on the right, you see the same chart for annotated tissue, or in fact, unknown tissue. And as you can see, the majority of the tissues at some point were unknown. Oh, by the way, it may be a little bit hard to see, but this is time, right? So the x-axis is time, and you can see how things progressed over time. Now, at some point, we try to buck this trend. We try to get rid of this unknown annotation of people not annotating. So remember that I talked about lowering the threshold for people, making it as easy as possible. So that's exactly what we did. We built a system, a software system that allowed people to annotate their data on their own computers very effectively, very quickly. So when we built that particular software, this indicated by the teal line here, you can see that it had absolutely no effect on the growth curve of this unknown data. And here the same picture. So making it easy for people, unfortunately, was not enough. I'll tell you how bad it got. So in this particular case of the instrument annotation, it was a drop-down box. So you know what a drop-down box looks like? It's a box with a triangular shape in it. And when you click it, a list pops out, and then you choose one of the entries in the list. And there were, if my memory is correct, there were 13 possible instrument names there that you should click one of. And the instrument that most people clicked was the top one in the list. And in fact, you can see that it was here, the ion trap was the top one in the list, but that was not the most popular instrument of the day. The most popular instrument of that particular moment in time was the Orbi ion trap. But they selected ion trap, but that was the first element in the list. So people couldn't even be bothered to scroll or look down 3 or 4 terms to find the correct thing. They just clicked the first one and be done with it. So it really did not help. It was really a big problem. But then, of course, you will have noticed that at some point this trend is going downhill. Yeah? So the unknowns are starting to be reduced. Why is that? Well, I call that affectionately the Attila effect, after Attila Czordas, who joined the PRIDE team at that time, and who then became the policeman in PRIDE. And his job was to look at all incoming data. And if the data would not contain a very, very minimal bit of data, so metadata, such as, for instance, the instrument name or the sample tissue of origin, he would reject the data set. So he, at that point, would just go back to the original submitter and say, I'm not putting your data in PRIDE because your data lacks very essential minimal metadata. And that had an amazing effect. Now, you have to keep in mind, this only managed to have an amazing effect because by this time, the journals were mandating deposition. So if we had done this 5 years ago, 5 years before, somewhere here on the timeline around 2008, it would not have worked because people then would have said, oh, so you want to make me do a decent annotation? I don't want to do a decent annotation. I'll not put my data in your database, bye-bye. So we couldn't force that. But by this time, the journals were now saying, if you don't put the data in the database, you don't get a paper out of this. And now we could police, because now there was pressure for people to do this. So unfortunately, the only way to make people annotate their data decently was to police them. And it still is the case to this day. Why does it go down in steps? That's because some of the data takes a little bit of time to become public because the publication process takes a while. So these are kind of leftovers, these last bars of unknowns. They're leftovers from the previous era. But it very quickly went to zero. And now anything that's submitted after 2013 will be annotated. But policing is important because people, unfortunately, don't like to annotate their data, which is a bit weird. I'll come back to that. If you think about what value there is in this data. Anyway, I've compiled a very small non-exhaustive checklist that should carry you forward at least a good part of the way to ensure the suitability of your data for archival efforts. So the first question that you should ask yourself when you annotate data is, have I explained or written in full all abbreviations? Abbreviations can be field specific. They can be very confusing for people. So make sure everything is always spelled out nicely. Second, are all metadata properly annotated? This is a really big thing, right? So have you provided the relevant information that people will need to reuse your data? This is very important. We talked about that. Third, are standard formats used to represent your data in? So is your data in some kind of weird binary format that nobody can read? In fact, is your data in Excel? If it is in the XLS format, just transform it into a CSV or tab delimited file, right? Don't use proprietary formats. I mean, the big question is 20 years from now, will people be able to read that file still? So if you use these binary proprietary files, there's a high probability your data will not even be readable anymore. So definitely try to use standard formats. And for many things, standard formats in text style exist. It's very easy to make them as well. Are your annotation terms derived from controlled vocabularies? So have you given them a fixed understandable name that is also put into context? Remember that it really helps with making your data findable. If any data is missing, have you explained the reason for why this data is missing so that people don't look at your data and say, oh, they forgot to upload run number 6 because I've got run number 5 and I've got run number 7. So I don't have the complete data set. But maybe there's a perfectly good explanation. Maybe run number 6 was completely botched because the instrument had a bad day and you had to throw it away. Then just write it down. It's kind of like the equivalent in a book where it says this page intentionally left blank. And data set 6 is not missing. Data set 6 was a disaster and that's why it's not included, right? So explain these kind of things. And then, am I using a trusted, long-lived third-party repository? Why is this so important? Because academic data sets and academic repositories on their own servers tend to have a half-life of 2 years. So we start today, anything that is not in a centralized repository, but is kept by people on their own, even their own groups servers, 2 years from now, half of it will be gone. Another 2 years, the next half will be gone. And very quickly, there will be practically nothing left. And the reason for this is very simple. Most people only start acquiring and making available big amounts of data if they're, say, in their second or third year of their PhD. But then, of course, they're close to finishing their PhD. So then they leave. And after they leave, nobody really cares about the data anymore. So as soon as something happens to the server or a hard drive needs to be replaced or something gets moved, this thing goes offline. And that's it, it's gone. However, if you would have put your data in one of the third-party repositories, of which many exist, and I've even shown you some, then you wouldn't have this problem. So definitely make sure to use a trusted, long-lived third-party repository when you make your data or your code or whatever available, right? That's why these things are there. They tend to be centrally funded and they tend to be capable of doing this. Now, you may also want to check whether you can share your data, your sensitive data with the world. So the first question is, if you're dealing with human beings, for instance, or data from human beings, do I have the proper clearances from the people participating in my study and maybe any other data originators to make the data publicly available? Because obviously, if I have patient data, I cannot just tell you where they live, who they are, and what problems they have medically, right? So this is a kind of a no-brainer, but nevertheless, it's important to point out. And you probably also want to make sure you know in advance exactly what kind of releases you want from them, because changing that afterwards is a giant pain. The problem is you have to track back everybody, make them come to your place again, and have them sign a new piece of paper. And people who don't want to do that, who you can't reach anymore, who can't be bothered, you lose all of their information for making it available. Then double check the clearances for sharing, for reuse, and for any combination of the data. There are very strict rules these days in Europe, this is the GDPR framework, through which restrictions are being put on this sharing of data that can be combined in certain ways. So definitely these days, double check. Don't be happy saying, oh yeah, this will be fine. Even better, get this to have this double checked by somebody else even. If it contains patient data, be very careful. If it's people, be very careful. Then, is the data properly anonymized, if at all possible, or at least de-identified to some extent? And so this falls within the same category. Usually you cannot make anything available without removing the personally identifiable data. That's where the problem with the combination comes from. Certain non-identifiable data can be combined with other non-identifiable data, and the combination is identifiable. And that's really a big problem because it's extremely difficult to predict beforehand whether or not that will be possible, because you may not know about other data sets that this data set can be combined with. So it's extremely difficult to mathematically prove that this is not going to be possible. In fact, it's probably impossible to prove, but the GDPR therefore says that you have to try your very best to ensure that this cannot happen. So that puts a lot of responsibility on your shoulders, but if you try to do a lot of effort to make sure that this is not possible, and some extremely intelligent individual manages to somehow make it work anyway, you could still get, you could still be okay because you've tried, right? It's something that you could have never imagined. So you're not supposed to cover all bases, even the ones that will be invented in the future, but you are supposed to cover the bases that are known now and that can be expected, right? So that's kind of the framework. Then, of course, as I said before, everything above, these 3 points above, should have already been sorted out before you see any people, before anybody participates in your study. And that's why we have ethical committees and other relevant experts that you can talk to. Don't wait with any of this. And then, of course, the last thing is, if you're sharing data with the world, you have to think about the license. You have to think about the legal framework within which this data can be or cannot be used or reused by other people. And we'll talk about that because these licenses are super important. So you may have noticed here at the bottom right of my slides, there's this little chunk and it says CC, a little guy, and it says by, and then it has an arrow folding in on itself and it says SA. And then it says that again in text underneath, CC by SA 4.0. 4.0 is the version of the license. CC is the name of the license, it's the Creative Commons license, as you can also see in the title. The little guy means, and the word by means author. So by whom? By me. And what does this by thing mean? It means that, and that's what's written here, you have to recognize me if you're going to reuse this stuff. So fundamentally, you have to do what I did whenever I took a slide from Dr. Masuzo that you've seen earlier. I put her name here saying, this slide is licensed as CC BY SA by Paula Masuzo, by the way, but it comes from her so that you know which slides are mine and which slides are hers. And so I give you her name so that you know that she made that slide. So that's quite important. And then the SA is an interesting thing. The SA thing says you have to share you have to share this data alike. If you want to reuse this data, so say you want to take a slide of mine and make your own presentation about that, you're very welcome to do that. You can do that, but the slide set that you make, which includes my slide, now has to be licensed under the CC BY SA 4.0 license as well. So this is fundamentally also the reason why I cannot make this thing available in another license, because the slides I took from Paula, these slides were already licensed with an SA clause. So I have to now, because I use her slides, I have to make my slides available under the same license. Yeah, this share alike system, which actually causes derivative works to be licensed under the same license, is known as an infectious license. It's a really bad time in the world to be talking about infectious licenses, but there we go, they're infectious licenses. And so whenever you reuse anything from a bit of work that is licensed under this way, it will infect your work. Why are these licenses so important that I've now been explaining? It's because data that does not come with a license, so you're too lazy to assign a license to this or you haven't thought about it, the default legal framework is you cannot share the data at all. So the author has 100% protection by default. If I would not have put this thing at the bottom and hadn't mentioned anything about this, then first, I would be in violation of the CC BY SA 4 because I use Paula's slides, but imagine that it's only my own slides. Then if you would take one of my slides and put them in your own slide deck and give a presentation on that, I could sue you. I could say, you don't have any rights, none at all, to reuse my stuff. So that's why it's so important to assign the right license. That's why it's the last point on my checklist. And when you license something, I think you should probably restrict yourself to these two licenses. Because this attribution license, the BY license, which mandates recognition of the original author, is how we do all of science. When you publish something and people reuse that knowledge or that protocol or whatever you developed or that piece of software, you would like them to give you a reference. Well, that reference is nothing but a recognition of the author. So this attribution license sounds like the default license in science. The attribution share alike one then is just a variation that says, if you're going to reuse my stuff, then you must make it available to other people as well. So you cannot take my stuff, make something new out of it, and that new thing is then only for you. You cannot do that. So I feel that what I gave you for free, you have to give back to me as well, and to everybody else, okay? So you have to think carefully about which of these two you would like to use, but one of these two will probably suit your purpose. So if you're thinking about how do these things combine with commercial interests, the attribution license actually makes it easy to make a commercial product out of this. Because all that, suppose somebody takes my slide and now makes a webinar about how to do open science and charges anybody who wants to see the webinar 50 euros. That is absolutely fine if I had used a CC BY license. Why? Because the CC BY license only requires them to put my name there and say this slide from this guy, this slide from this guy, this slide from this guy. But the product that they make, the webinar, the video that they make of this, they can do with what they want. So they can sell that to people and it's fine. However, since I'm using the CC BY SA license, if they do this now, they can still sell the video. There's no problem in them selling the video. The problem now is that if they sell the video, they also must make that video freely available. So while they must make you, they could make you pay for the video, they should also offer up a free version. So this attribution share alike thing makes it harder for people to claim some kind of exclusivity on your work downstream, to add a small amount of value and then make this whole thing commercial, whereas the CC BY allows that fine. Most companies don't like this share alike because they're very much afraid that anything that shows up in their work, which is a share-alike licensed item, is going to make everything that they do freely available to anyone. So usually this is kind of an off-putter for companies. So that's one of the things you need to think about. I mean, if you want to build a company yourself, you have to be careful on your own work if you use the share-alike, for instance, right? So be careful and think about this, because that's why this is in red. It's an infectious license. It has repercussions downstream. There are also, and some of you may know of this, other CC licenses that I don't really talk about. The first one of these is the CC0, so the number 0. And CC0 is essentially a public domain license. So CC0 is, this stuff is now available to anybody without any restrictions whatsoever. It is what all copyright defaults to at the end of their copyright life cycle, but a copyright life cycle is ridiculously long. So it's up to 60 or so years after your death, I think, in most places. And there's this running gag that says that the copyright extends for as long after your death as Mickey Mouse and Walt Disney, right? So Disney doesn't want to lose the copyright on Mickey Mouse, so they lobby like crazy to make sure that it is extended since the death of Walt Disney forever. And funnily enough, it actually does track that to some extent. So it's a bit weird that way. The other license that exists in this Creative Commons library is the non-commercial. The non-commercial license prohibits any downstream commercial uses. But the problem with this license, and the reason why I don't recommend it, is that commercial uses are extremely broadly defined. So people who try to do, say, automated text mining from research papers and purely for research purposes, they're also inhibited by the non-commercial license, because the interpretation is any useful product downstream that could potentially have commercial value. And so if somebody would mine all papers for, say, protein-protein interactions, and anything that is non-commercial licensed would create a problem, because even though they would give away for free the protein-protein interactions, somebody else might at some point sell those protein-protein interactions as part of a larger compendium, and that is not allowed. So the non-commercial thing, it's not a very elegant license. I would try to stay away. Make a share alike, it probably has the same effect of dissuading companies to use your material for ridiculous commercial gains. And then there's also a non-derivatives license, ND, but that kind of is a silly thing. It says, I am licensing my work, but you cannot reuse it. So you can look at it, you can print it and hang it in your room, but as soon as you make a derivative of it, if you ever use it for anything, oh, but that's a big no-no. You cannot use it anywhere else. That's kind of very restrictive. So then I think the whole Creative Commons, the Commons thing is gone. It's creative, my own stuff. So I don't think a Creative Commons ND license is particularly useful for scientists. So that's why I keep it to these two. Now, very important caveat with all of this. Creative Commons licenses are for content. They're for paintings, books, presentations, manuscripts, all these kind of things you make, but not for software code. Software code is special. Software code has its own licenses, which I'll come back to later on when we talk about open code. And Creative Commons is not just some crazy idea by a bunch of weirdos. It is actually used all over the world in lots of stuff. And the most famous example is Wikipedia, which is essentially completely built on Creative Commons. And you can find out more about Creative Commons in these two URLs at the bottom. Right. So let's talk about open access now. So the publishing world. The first thing you probably want to know about is that the publishing world is a very interesting world. Homework for tonight, go on the Guardian website. You can see the URL here at the bottom. It's a text from 2017 or an article from 2017. It's called The Long Read, but it will probably not take you more than half an hour. And look at the title. It says, is the staggeringly profitable business of scientific publishing bad for science? It says here, profit margins to rival Google. It actually turns out that in 2010, Elsevier actually had a higher profit margin than Apple, Google, or Amazon in that year. So the publishing business is a weird business that makes a ridiculous amount of money, and the way it was built, and this is nicely explained in this article, the way it was built is really nasty. It is built on an exploitation of a personality trait, a weakness in the personality traits of scientists, which is that scientists are heavily ego-driven. And the whole publishing business of the high impact factor journals is built around this desire to be elitist, to stand out from our colleagues, to be better than everybody else, and make us pay through the nose for that. After you have read this long read, you will never look at scientific publishing in the same way. So it's definitely important to read that. It's very, very well worth your time. Anyway, open access. What does open access mean? It means that we're no longer going to pay a journal to get access to a piece of work, to an article, as readers. Yeah, so the reader is not paying, but somebody is paying somehow. So the first way to make sure that everybody can read your paper for free is the so-called gold open access. And you can take the gold thing literally, because this means that as an author, you are now paying. So you have to pay the journal an additional amount of money in order to make your article available for free. Essentially, you're paying all the costs, all the profits, sorry, you're paying all the profits that they're not getting for making your thing available for free. Importantly, in most journals, the other papers that come out in the same volume or the same issue of that journal will probably not all be open access. So maybe there's 5 open access papers in Nature this month, and the other ones are paid for, right? So you cannot read them. So libraries will continue to need subscriptions to these journals in order to read all the content in these journals. So that translates into a neat double profit for the journal. So 5 or 6 people are paying them roughly 3,000 euros. That's roughly the standard price, 2,000 to 3,000 euros before tax, to make an individual paper open access. Meanwhile, they're also getting all the subscriptions they would otherwise get anyway. And as a result, they're earning double profit. That is something that rubbed a few people so very much the wrong way. And we talked about this, that the Bill and Melinda Gates Foundation, as well as the Wellcome Trust, have said, that's enough, basta. We're not going to allow this to happen anymore. We are going to say that any journal that uses this mechanism of making some papers open access because you paid while everything else has to be subscribed to, we refuse to let our funded scientists publish in such journals. Either the whole thing is open and everybody can read everything for free in your journal, even though the authors might have to pay then, or we don't publish in it anymore. So this is a model that is very quickly getting outdated. A much nicer model, and this is the model that my group uses nowadays as much as we can, is green open access. And green open access is based on the fact that you write your manuscript. So the initial copyright on the thing that you have written lies with you. And that thing you can share for free with the world because it's yours. You can make that decision. Very often when you do this, you're asked to assign a license, and the license tends to be Creative Commons by or Creative Commons by SA, right? So the 2 licenses we talked about. Now, the thing is, why can you do this? Well, because you write the manuscript. Why can you not do this with the final version of the paper once it's printed in the journal? It's because the journal has now added value that they have ownership over. And the added value is the process of peer review that they took care of for you and the typesetting. And so the final formatting. So that is something you don't own, and therefore they can claim that you cannot make it available for free. But the Word file or the text file that you wrote up before you submitted it to the journal, that is yours and yours alone. And this whole green open access thing is based around that. So just chuck that manuscript into a public repository, and many exist, and the most important one for the life sciences nowadays is bioRxiv, and just have it freely available to everyone at that moment in time. A lot of these journals in the beginning tried to stop this from happening, and they used a very nasty trick. And the trick was, if you submit your preprint, your manuscript, to bioRxiv or archive or something like that, before you submit it to our journal, we will reject it anyway. So we will not consider anything that you've already made publicly available. They have beautiful explanations for this, but they only essentially talk about one thing at the basis, and that is, this is killing our business model. So nowadays, there was a lot of pushback over the past years. So nowadays, every journal of any repute will allow you to deposit in bioRxiv. The chemists, as far as I can tell, in the broader life sciences like world, so chemists are kind of adjacent to that. The chemists were very, very late to this game, but even they have now changed their policies. So the Royal Society for Chemistry and the American Chemical Society, probably two of the biggest in the world, they now allow for all of their journals to do preprints. So preprints are now everywhere. There's also a third method, which is delayed open access. This is actually something that was, I think, originally negotiated by the NIH, and they essentially mandated that the journals could have an embargo period of, say, 6 months to a year, within which the paper would not be publicly accessible, so you had to pay for it. But after that year, the paper would become publicly accessible. So the whole idea is, when the paper is hot and new, you have to pay the journal to see it. Once the paper is already a little bit older and people don't really care about it anymore, yeah, then they give it away for free. So it's kind of like a patent where you have a certain period in which you can exploit it exclusively. This is kind of like the same system for journals. Now, in Horizon 2020 projects, and this is probably relevant for you guys as well, you have to make everything open access. And it's actually policed, right? So they actually check all of that. So if you want to make your work open access, what's the best way? Well, the best way is to go green open access. Just chuck your stuff at bioRxiv. You get a DOI, you get a link. Later on, it will be linked to the publication if it gets accepted in a journal. So even that link is there, but go bioRxiv. Funnily enough, the embargo period will not work for you because in the life sciences, the vast majority of embargo periods are 1 year and the EU mandates 6 months. So they're very clever. The EU, or the European Commission rather, said, we want this thing public in 6 months, probably knowing full well that most journals use 1 year. So they're forcing you not to play this game. They're forcing you to either do this or that, but the gold open access just doesn't make any sense anymore. Go green. You can actually easily find out about journals that are completely open access because there's this brilliant website called Cofactor, and you can see the URL at the bottom. And this journal allows you to, this website allows you to select a journal based on subject, whether or not they do peer review, what kind of open access they offer, how fast they are, and any other criteria that you may have. And then you can click search and you can find out which journal could suit you. That's if you want to go and find a journal. The alternative is that you use Sherpa Romeo, which is an online tool. You can see the URL here at the bottom, that allows you to find out what the policy is for open access for a given journal that you're interested in. And apparently they have a new version. I only saw this now after I made the screenshot. So there is a HTTPS version 2 Sherpa Romeo, and I'm sure they will forward the old link to the new one. But the nice thing here is if you already know which journal you want to publish in, or maybe you know the 2 or 3, so you're not looking for a journal you know, then you can find out whether or not it's possible to say use green open access. And these guys will tell you whether that's possible and what the embargo period is and so forth and so forth. And you can see what their mission is, opening access to research, and they're very useful for that. Green open access, as I mentioned, it really is the way to go. And there's very many ways to do that. So probably the most popular one in the world used to be arXiv, and this is where the physicists and the mathematicians put their work. Now we have bioRxiv. Here it's still beta, but it's not in beta anymore. It's very popular nowadays. So for life sciences work, that's where things go. Another one that I really like is PeerJ preprints. That's actually where we put the Do You Speak Open Science manuscript that I talked about at the start. I like PeerJ because they have a wonderful interface. It's one of the best designed ways of doing these kind of websites. bioRxiv is pretty cool, but it's pretty basic. PeerJ has a nice vibe to it. They both work extremely well. There's some interesting information as well on innovations in peer review. We'll come back to that. It's an interesting topic in its own right, and it's kind of linked with open access and green open access, but not completely. So I'll come back to that later. And then, of course, there's also institutional repositories. So we have one at Ghent University where you can upload all your papers, and even in Belgian law, but this is Belgium. In Belgian law, we now have a clause that states that we can definitely make our final accepted version of the manuscript before typesetting. We can make that available for free. So we can take the final accepted version, even with peer review included. We can chuck that at our institutional repository and make it available to the world. So we are covered by our local law, apparently, to do that now since 2 years or so, which is a fantastic development because it shows that our government too really cares about open science. And also, they're also sick and tired of paying publishers large amounts of money to make the research available. It's all taxpayers' money and it just ends up in big corporate pockets, unfortunately. And of course, my group does practice what we preach. And in fact, everything we do nowadays goes to bioRxiv by default. So as you can see, if you search my name, you can already find 37 papers. And we're going to continue to do this for everything that we do. It also has a really nice benefit because as soon as you chuck it at bioRxiv, you get a timestamp from bioRxiv. So you can say, I submitted, for instance, this paper on 2020 on the 14th of January. So if you ever want to win a Nobel Prize, it's very important that you're the first with something. And in the past, they would look at the submission date to the journal. Nowadays, you can have the submission date to, say, bioRxiv. So you can bring out your work pretty quickly, tweet about it, then lots of people can go and read it. And actually, this happens a lot. A whole buzz is generated even before your work gets peer reviewed. And it's quite funny, sometimes the peer review starts already with people commenting on your paper saying, oh yeah, this is very interesting. However, would you not have better done this or that? It's not that common yet, but it happens. And it's actually a very interesting development as well. Okay, now that's as much as I will tell you about open access. Time to talk about open code. So I told you that the manuscript that you write is probably the least interesting thing of your entire work. The data is probably the most interesting bit and the thing that will live longest. Another way to formulate this, and I stole this slide again from Paula, she has a very good way of putting this. Your paper is essentially the advertisement for your data, for your processing, for whatever conclusions that people could get out of this. Because the code on GitHub, together with the data, I add myself, actually represents the research performed. And in the life sciences, this is increasingly the case, right? Increasingly, the work is not necessarily done in the wet lab. The work is done in the wet lab combined with the bioinformatics afterwards. So without the interpretation, the data doesn't really mean anything. And the way that the data interpretation is done is therefore quite crucial. You need to know whether this is all done right, and you need to be able to play a little bit with this. And nowadays, it's actually become very, very easy to actually make all of the code that you have written available and all of the plots and generate everything on the fly again so that everybody can reproduce very nicely what you have done, how you have done it, and can actually use this as a basis to do new stuff because they can actually follow from your code and tweak things and make things better. So there's a consortium called NumFocus that really try to push open code and therefore also allow better science. And they do an amazing amount of things that are super relevant for scientists, and a lot of things we use a lot in the group. And one of the key things that they've done is Jupyter Notebooks. And Jupyter Notebooks, I've shown you one on the previous slide. This is on GitHub, but it's a Jupyter notebook. You can see that here in notebooks. And it's a mixture of comments, code, and output. And so it's a great way to put your entire analysis pipeline, which you normally would chuck in a script somewhere and lose on your hard drive in 700 different versions, in a well-documented overview of exactly what you have done, how you have done it, and what came out of it. So it's a brilliant way of documenting your code, and it's a great way to communicate with other people because you can make this notebook available to everybody else, and they can run it and analyze it. They can run their own data through your analysis pipeline if that's relevant, or they can tweak your analysis pipeline on the original data. So these Jupyter notebooks, here's some more examples, are really, really interesting, really useful, very powerful. They're also brilliant teaching tools, so we use them as teaching tools as well. And fundamentally, it brings your development process, your code sharing, and your reporting on your work all together in one place and in one effort. You do a good job in making this thing from the start, your code sharing is already done. If you chuck all of this at GitHub, then everything is already archived, everything is already available on a third-party site. It's a brilliant way to share your code, especially if it's mostly script-based code. If you're building very sophisticated applications, that's a different thing. Then you really want to go into the traditional code availability. But for many of you, it will be scripts, and scripts are perfect to be shared in this way. Also, you can consider, and this is a nice point here, that a Jupyter notebook becomes a publication. Because what else do you need, right? If it's the data, if the data is described, if it's in the right format, if you have all the analysis and you have the text around the analysis that interprets what we see, then fundamentally you have something that is as close to a publication as you could possibly get. So who knows, maybe 10 years from now, a Jupyter notebook will actually qualify as a publication. It's currently not the case, but it's not unthinkable that this might happen. Okay, as I mentioned GitHub already. Open code can be shared and managed through third-party systems, and probably the most popular one is GitHub, although Bitbucket tends to have a few adherents. You can do a lot of stuff in GitHub. The cool thing is it doesn't just have to be code. Do you remember this title? This thing is the manuscript I showed you at the start. This manuscript was completely written in GitHub. So it is a piece of, if you want, book code that is available on GitHub. So GitHub is being abused for many things nowadays, for courses, for code, for books, for text, for manuscripts. So it's a great way to keep an archived version of everything you've ever done to a thing. And so in this case, it's a manuscript proper. So GitHub is very, very interesting. It's a third-party site. It's kept for you. It's archived for you. It's very useful, not just for code. Now, what is also interesting about a system like GitHub is that it's actually a social system. So people can actually like or star your work, and people can fork your work. And when somebody forks your work, it essentially means I want to take this and use it in my own code. I'm going to split the work and take it and use it myself. And the system keeps track of how many people liked your work and how many people forked your work. So you can imagine that on your CV, you say, I've made this particular piece of code and it's been liked or starred 400 times and it's been forked 3,200 times. So look at the amazing impact that I have had on the field. And remember what I said at the start about open science, that it's really cool when people build on top of your work. I mean, there's few things more fun than seeing other people you have never heard of forking your code and being happy by starring your code. That means that you're doing something worthwhile, right? And it's something that you definitely can put on your CVs. So it's something to actually embrace very actively and do things with, because it will give you credit, it will show off your capabilities, it will show off your value, and it will allow you to help other people at the same time. There's very little opportunity for losing here. The other thing that people can do, and this happens to our code quite a bit because everything we do is on GitHub, people do pull requests, which essentially they take a version of your code and then they find an issue, they fix the issue, and then they send you back a fix and ask you to merge that fix back into your code. So other people are helping you fix problems in your code, or are helping you. The issue could also be an extension, right? Would allow you to do something you were not capable of doing before, and they just contribute to code and it goes back into your system. So now you have a better system. This happens actually reasonably often for us, and it's amazing every time it happens. When out of the blue comes this email where somebody says, oh, by the way, I did this thing, can you merge this back into your code so then you have that available as well? It's brilliant. So it's a fantastic way to work together with other people. So yeah, GitHub is definitely interesting and definitely useful. And as you can see, we used some pictures here that came from GitHub, and thank God they're licensed in a way that we can reuse them. Now, we talked about licenses for content. Now we talk about licenses for code. So I'll show you the key licenses here, just like I did for content, and there's essentially 3 that make sense. The first one is the MIT license. The MIT license, as you can see from this header on this website, choosealicense.com, is essentially, you know, do whatever you want. I made this thing, here it is, do whatever you like, just let people know that I did this. And don't come sue me if something crazy goes wrong. If you use my code in a nuclear reactor and the thing blows up, it's not my fault. I didn't tell you that this was okay. So it's very, very easy to give the code away and to have people use it. And all you want is a reference, a call out, like this code was done by that guy. If you're concerned about patents and people patenting stuff, you want to enable people to patent stuff, then the Apache License 2.0 is a good idea. This is what my group runs on because we think all our stuff should be compatible with businesses. If companies want to make use of our code, that should be possible because they pay taxes too, you know. Also, if we ever want to build a company, we may want to build on our own code. So we use Apache 2 because it's more permissive towards companies. And then the final thing is the GNU GPL license. That's a weird one because it's like the CC BY-SA, the share alike. This is the original infective license. And in fact, it was named a copyleft license rather than a copyright license, just to show you how crazy a license this is. So the GNU license says, if you use any line of my code in any other product, that product needs to become GPL licensed as well. So companies avoid GPL licenses like the plague. There's some high-profile cases, one involving Cisco systems, where they apparently had used bits of GPL code, I think from the Linux kernel, and they really were hammered in court. And ultimately, they settled out of court, but it mustn't have been easy for them because they were caught red-handed in using GPL code in stuff that should not have the GPL code. So companies really don't like that. If you want to make sure that your code is always free and anybody who does anything with your code will remain free for everybody for all eternity, use GPL. I originally used GPL, and then later on I thought that's actually not nice towards companies because I don't really want to stop them from doing things with my code because, again, they pay taxes too. But that's my personal take. If you have another take, it's all fine. With these 3 licenses, you should be able to do whatever you want. There are many more licenses. And you can see here, I want more choices. That's fine. You can have more choices, but really I would stick to these 3. If these 3 don't do what you want, you're probably making it too hard on yourself. Unless you're a company, but then you usually make your own license and you hire a lawyer to do that for you. So a brief summary again, you have the GPL, which is this copyleft thing, which is infective, like the CC BY-SA. You have a half version of that, which is the limited GPL or the linking GPL, depending on how you mention this. And here, this is about non-infective linking. The idea is if you compile my code and you use my compiled code as, say, a library, then you don't have to use the GPL license. There is even a special version for Java developers. It's the GNU GPL class path license, which is kind of the same. It says if you take a compiled jar file and put it in the class path, that doesn't make the other thing GPL. So these things exist, but to be honest, if you're going down the path of an LGPL, you may want to consider going Apache 2 or MIT because it's probably closer to what you really want. As I said, Apache 2 is the best one for companies. MIT is highly permissive, but for companies and patents, it might be a problem. And then BSD is an even more permissive license that essentially says, here's everything, do whatever you like. And so these are the most common ones that you will encounter. Again, I recommend looking into GPL, Apache 2, and MIT. One of these 3 will suit you, probably. And of course, there's a lot of info on the web from these 2 links if you need more. Interesting point. If you want to change your license in the future, you have to be very careful because most of these licenses have a license changing clause that states that every contributor to the code ever has to unanimously agree to changing the license. So as I said, I used to have GNU GPL and then I changed this. And I looked up what I had to do to change it to Apache 2. And it turns out, yes, I need the full agreement in writing from everybody who ever did anything on my code. If they put a comma in another place in a comment, they technically have to write written consent. Now, I was lucky at the time, it was me and a master student, so that was easy. I still have all of that paperwork actually, so to make sure that I can prove it. But imagine you've got a project that's been running in a group for 5 or 6 years, where a lot of students and master students and PhD students and postdocs have come and gone. They've all touched the code one way or the other. Now they've all dispersed again, and you say, ah, I want to change the license. So first, have fun tracking everybody down. Second, have fun getting everybody to reply. And third, what if only one person there either dislikes you or has a bad day or really doesn't like the new license? You're in trouble. Yeah? Because if they say, I don't want to do this, that's it, you cannot change it. So think carefully before you start your project to make sure that the license that you choose will last you for the lifespan of the project. As a result, personally, for scientists, I tend to think Apache 2 and MIT, one of these two are best because they don't box you in. Yeah? But again, your mileage may vary. Right. So last bit is about open peer review. This is a short bit because I don't have too much to say about that. Open peer review is a good idea. Yeah? And if you want to know more about this idea, there's this brilliant manuscript paper here that was published in Faculty of 1000 Research. And you can remember this person maybe, Paola Matsuzo was a co-author on that. Here is the link, by the way, it's open access, of course. And it talks about how peer review could change. Now, what I want to do with this is I would like to showcase things that are good and bad about the plans that people have on this. So the idea behind it all is open peer review. And you can have open peer review in multiple ways. You could say, from now on, all the reviews, anonymous reviews, are posted alongside the manuscript. That's kind of semi-open peer review. You can also have open peer review in which you say, now all the reviews are posted alongside the manuscript, but we put the names of the people who did the reviewing. So as you will find out, I'm not a big fan of open peer review. There's a reason why peer review is anonymous. Yeah? And it's right here on the top. Junior researchers, so PhD students, tend to do most of the peer reviews in the world. And unfortunately, junior students are in a very weird power dynamic. Essentially, all other scientists have more power than them. So if you are a PhD student and there's this big professor's article or manuscript and you're asked to review it and you say, yeah, but this is a really bad article, I really don't like this, and you write a pretty to the point review that says this is wrong, that is wrong, this is wrong, that is wrong, I'm sorry, this is not publishable, and it has your name under it, now that very big powerful scientist could really ruin your career. And I know a lot of big powerful scientists. Not all of them I would trust with my name if I write them a bad review, right? Especially not if I were a PhD student, because unfortunately we have a power dynamic. We have a very, very strong power dynamic in academia. It creates a lot of problems outside of open science. We have a very strong power dynamic. And the problem is we have a lot of quite inflated egos. So you don't want your name under a negative review. Of course, if you're going to write a review that says, yes, publish, most amazing thing I ever read, I wouldn't mind putting my name there because now it might work in reverse and people might like me now. But fundamentally, don't do this. Keep it anonymous so that people can be fair and people can say what they think. This is, to me, very important. There's also post-publication peer review. So when people can write comments with manuscripts, and PubMed has experimented with this for many years. The problem is that if you look at it, there's something wrong in our psychology as humans. We tend to have predominantly negative comments on these papers. It's somebody who's peeved about something and then goes and writes a really negative review, sometimes even a personal attack if somebody felt slighted by a manuscript. You very rarely find constructive, positive criticism. I don't know why, maybe it's something in our psychology. We read something we like and say, oh, this was great, I like it. We don't go online and type, hey, this was great, I liked it. It's something that somehow doesn't really work very well. So post-publication review, yes, it's not a bad idea. Unfortunately, most people dislike it because they only get shouted at online, or even worse, somebody on Retraction Watch or so starts tearing your paper apart. It's a really good concept, don't get me wrong. The implementation, however, seems to lack right now. Something that you may have noticed is that when you do peer reviews, you get nothing back. So we're all peer reviewing for free. Journals are making a lot of money out of that. And at the end of the day, nobody's going to ask you, so how many papers did you peer review, and use that as some means to evaluate you. If you want to get your PhD, nobody's going to tell you you need to have peer reviewed 7 papers. They will tell you you need to publish X many papers, but they won't ask you how many did you peer review. If you write on your CV, I do peer reviewing for this journal and this journal and this journal, people will just say, oh, okay, great, they do peer reviewing. But they will not really use this as a means to say, oh, they do peer reviewing, now we're going to give them a promotion, or we're going to give them a position. So today, you don't really get a lot of credit for peer reviewing, whereas it's a fundamental task, of course. And it's an important task. So the manuscript, or the paper I just showed you on the previous slide, that actually has some pretty good ideas on how you could potentially extract credit from peer reviewing. And a lot of people have tried, but so far it hasn't really taken off. So maybe if somebody has a brilliant idea, you might change the world that way. There's also an interesting question. Who owns peer review? So I don't know if it's ever happened to you. Maybe you guys haven't been around long enough, but I have. And I've had reviews where I thought, yeah, I would like to write a blog post about this review, either because it was an amazing review and this is how reviewing should be done. Even if the review rejects my paper, it can be amazing. I once had an amazing review that was 4 pages long that rejected a paper of mine, and I finished reading that, and I was like, wow, you know, bravo, this is a fantastic review. It's bad for me, but it's a really good review. So you really want to write something and say, look, this is how it's done, people. It's really brilliant. So I ask every editor that I meet, can I make somebody's peer review of my paper publicly available? Because I didn't write this, right? So the original author is the reviewer. And there's no license associated, as far as I can tell. So technically I cannot do anything with it. And so far I haven't had a good answer on this. People essentially say, we don't know. We don't know whether a peer review text can be made publicly available, which is really bizarre, right? I mean, that really stands in the way of open peer review. If I, as a journal, want to publish somebody's peer review alongside the original manuscript, then I need, of course, to have the copyright on that, or at least have the permission like a CC BY, or rather in this case, a CC 0, because you don't want to have a by, you don't want to have an attribution. But apparently, classical journals don't have a clue, and they don't have a system in place. PeerJ, which I mentioned earlier, they have a pre-submission server. They also have a journal, and they actually publish every review. But I think they do it anonymously, unless you really want to be known. You can choose, I think. But they actually publish all the peer reviews, and for sure they then have a system to license that peer review, so that you cannot claim that it's a violation of copyright if they publish your peer review, because you agree to the license when you do the reviewing. So this is something that I would hope journals sort out. Make sure that everybody knows that there is a certain license and that this peer review is not yours as soon as you've written it, because you're actually asked to write it by somebody for something, and they can actually put that rule down for you. Okay, so in the end, I do think that the medium level of open peer review makes a whole lot of sense. I actually think that the fully closed peer review, where the scientific community never gets to see the peer reviews, I think that's bizarre. Why is it that we cannot see the reviews that have been written during the process of getting a thing published? I mean, sure, I've been an editor, I've been a reviewer, I've been an author. I've seen some pretty nasty, bad, horrible reviewer comments. But to be quite honest, do we need to shield people who write really bad comments? I mean, first and foremost, they're anonymous, so nobody will find out who they are. And second, I mean, it's honest to show people how the peer review works. And sometimes somebody writes a really bad review. Bad, and not in the sense of it's negative, but bad in the sense of it's a useless review. Very often, however, the reviews are actually very insightful, and they show how the process went from the original manuscript to the final version, or they show the contribution of another scientist who says, try this idea and it turned out to be amazing. Right now, these people don't get any credit. They will still not get any because their name's not associated, but at least people will figure out that, oh, somebody else gave them that idea. So this kind of thing, I think, is very important. I think it's very useful to find out what the peer review looked like on manuscripts. And I would hope that in the future, in the not too distant future, this will become much more commonplace to actually publish the reviews that people make. I don't see a problem with that, but I still think they should be anonymous, right? You should be able to write your review without having to worry that somebody will come and kick you in the shins because you were nasty to them. Right, that's me done on this aspect on the open peer review. But again, that manuscript is super relevant that I highlighted if you want to know more. Final thing, final 2 slides, call to action. So a funny thing happened to me a few years back because a sociologist wrote an article on some work that was done in my group. And you can see the reference here. It was Mackenzie and McNally. So Ruth McNally was the person I talked to in Theory, Culture, and Society in 2013. And I was really afraid of this because I had no clue what they would write. I mean, a sociologist, I have no understanding of sociology at any level of depth. So what the heck am I going to expect from this? But they actually did a really good analysis of the kind of stuff that we did. And it's relevant here because what we did was built on open science. And so they say, and these are literal quotes from the text, that's why they're quotes. This desire to reactivate data, so that was public data, is widespread, and Klee et al., so Klee was a student who did the work and was the first author, are not alone in wanting to show that far from being places where data goes to die, which is something that we actually put in our article, such data collections can be mined for valuable information that could not be obtained in any other way. This is a key take-home message. Open science means that there are now many such data collections, which you can mine for information which is valuable and not available in any other way. It is open science that allows you to do valuable, revolutionary research. Not my words, their words. Oh, they say people try to reactivate sedimented data. I only put that there as a joke. If you've ever been to a water treatment plant where they reactivate the sediment, it stinks really badly. So it was quite funny to think of us being the stirrers in the sedimented tank, doing the dirty job, really. Then these things that we're doing are experiments in seeing. This is about the visualization that we do in furnishing ways of seeing how data on proteins, because that's what we did. We looked at mass spec proteomics data, could become reusable, could be reactivated. There's that word again, reactivate, reactivate, reactivate. And now we've become socialist, collective property rather than the byproduct of publication. So this is the thing I said several times, is that your data is more valuable than your paper. In the past, people used to think the data is the bycatch, the byproduct of my amazing publication. Nowadays, I would say the publication is the bycatch, is the byproduct of the real thing, which is the data. And this data is not your data. This data should really be collective property because it was collected with public funded money. And as a result of making your data part of that collective property, you enable this reanalysis in interesting new ways that were not possible before. So in these 3 short paragraphs of text, the sociologists summarized perfectly what I think is the main tenet of open science these days. Why we care about this, why you should care about it, and why you should be proud of your data, why you should take care of your data, and why you should be responsible for getting your data out there, and at the same time learn how to reuse other people's data. So, parting message. Open science, actually, think about it, is a treasure of unlimited size. If I take from the open science data pile, the data will still be there for you to take from. We're not in each other's way. And the big question will be, what will you do with this endless treasure? Okay, that's the whole big question. So with this, I am done. these are all the people in my group. And here, Paola Mazzuto, as you can see, she gets a highlight because I used some of her slides. Not in my group anymore, so she moved on, but she's here and she's watching over us. Okay, thank you very much. And I'll now happily take any questions that you guys might have.